The Hepatic Apocalypse, 10 Dietary Assassins Fueling Liver Cancer. The liver is a biological fortress, tirelessly filtering poisons from your blood. Yet even this resilient organ possesses a breaking point. We are witnessing a terrifying surge in hepatocellular carcinoma, frequently fueled not by genetics, but by the toxic compounds hidden on our dinner plates. These are not just empty calories, they are metabolic disruptors, capable of scarring tissue and mutating cellular DNA. To protect your longevity, you must identify the enemies camouflaged as sustenance. The investigation begins with the most socially celebrated carcinogen in human history: alcohol, especially heavy drinking. To understand the catastrophic impact of alcohol on the human liver, one must first dismantle the cultural romance that surrounds this substance. Ethanol is the most socially accepted psychoactive drug in human history, woven into the fabric of our celebrations, our religious rites, and our mechanisms for coping with stress. It is the champagne toast at a wedding, the beer at the ballgame, and the whiskey poured after a long week. Yet beneath this veneer of social lubrication lies a chemical reality that is biologically devastating. When we ingest alcohol, we are essentially drinking a solvent, a volatile organic compound that the body recognizes immediately as a poison. The liver, in its role as the body's primary filtration plant, takes the brunt of this assault. While the brain enjoys the transient euphoria of intoxication, the liver engages in a desperate, high-stakes biochemical war to neutralize the threat before it destroys the organism. The correlation between heavy alcohol consumption and hepatocellular carcinoma, HCC, the most common form of primary liver cancer, is not merely statistical. It is a direct, causal, and mechanically understood pathway of destruction that has been mapped by pathologists with terrifying precision. The biological tragedy begins with metabolism. When ethanol enters the bloodstream, the liver halts its other vital functions, processing nutrients, regulating blood sugar, producing bile, to prioritize the elimination of this toxin. The primary mechanism for this clearance involves an enzyme called alcohol dehydrogenase, ADH. ADH oxidizes ethanol, stripping it of hydrogen atoms and converting it into acetaldehyde. This transition is the critical moment of danger. Acetaldehyde is not a harmless byproduct. It is a highly reactive, toxic carcinogen capable of causing significant damage to the structure and function of cells. It is estimated to be up to 30 times more toxic than the ethanol itself. In a healthy scenario involving moderate intake, acetaldehyde is quickly further metabolized by another enzyme, aldehyde dehydrogenase, ADL, into acetate, a relatively harmless substance that is eventually broken down into water and carbon dioxide. However, in cases of heavy drinking, the liver's enzymatic capacity is overwhelmed, the assembly line clogs. Acetaldehyde accumulates in the liver tissue, where it begins to wreak havoc at a molecular level. It binds to proteins and DNA, forming bulky adducts that interfere with DNA replication and repair. This is the genesis of mutation. By physically damaging the genetic blueprint of liver cells, ethanol acetate sows the seeds of cancer, creating errors in the code that controls cell division. However, the carcinogenic nature of alcohol is not limited to the direct mutagenic effects of ethanol acetate. The process of breaking down alcohol generates a massive surplus of reactive oxygen species, ROS. These free radicals are chemically unstable molecules that strip electrons from other molecules in a frantic bid to stabilize themselves, a process known as oxidative stress. In the context of the liver, oxidative stress is akin to a biological wildfire. It damages lipid membranes, denatures proteins, and further shatters DNA strands. To compound the injury, the metabolism of alcohol consumes the liver's supply of antioxidants, specifically glutathione, leaving the organ defenseless against this internal combustion. This state of chronic oxidative stress triggers a profound inflammatory response. The immune system, detecting cellular death and damage, floods the liver with cytokines and white blood cells. While inflammation is a healing mechanism in the short term, chronic inflammation induced by habitual heavy drinking becomes a driver of disease. It creates a microenvironment that promotes cell survival over cell integrity, encouraging damaged cells to proliferate rather than die, a hallmark of cancer development. The journey from the first drink to liver cancer is often a slow, agonizing progression through several distinct stages of pathology, beginning with hepatic steatosis, commonly known as fatty liver. Alcohol alters the liver's lipid metabolism, causing it to synthesize and store excess fat. Under the microscope, the hepatocytes, liver cells of a heavy drinker appear swollen, distended by large vacuoles of fat. This condition is reversible with abstinence, yet it is the canary in the coal mine. If drinking continues, the condition progresses to alcoholic steatohepatitis, ASH. Here, the fat accumulation is accompanied by widespread inflammation and ballooning degeneration of liver cells. The liver becomes tender and enlarged. This is the tipping point. The chronic injury activates hepatic stellate cells, which are normally quiescent and responsible for storing vitamin A. Upon activation, these cells undergo a phenotypic transformation into myofibroblasts and begin to pump out collagen, creating scar tissue in an attempt to patch the cellular wounds. This process is called fibrosis. As fibrosis advances, the liver's architecture is fundamentally redesigned. The soft vascular tissue is replaced by bands of tough, fibrous scar tissue. This is cirrhosis, the end stage of chronic liver disease and the most significant risk factor for liver cancer. In a cirrhotic liver, the flow of blood is impeded, creating high pressure in the portal vein, portal hypertension, and forcing the blood to bypass the liver entirely, allowing toxins to circulate freely to the brain. But within the liver itself, the situation is even more dire. The remaining healthy cells attempt to regenerate, forming regenerative nodules surrounded by scar tissue. These nodules are hotbeds of cellular turnover. In this chaotic environment of rapid division, oxidative stress, and impaired DNA repair, the likelihood of a malignant mutation skyrockets. The cirrhotic liver is essentially a precancerous organ. Once cirrhosis is established, the risk of developing liver cancer increases exponentially, with studies suggesting that a significant percentage of heavy drinkers with cirrhosis will eventually develop HCC. The definition of heavy drinking is a subject of much debate, but the epidemiological data suggests that the risk is dose-dependent. There is no clear safe lower limit when it comes to carcinogenesis, but the risk escalates dramatically with volume and frequency. Binge drinking, defined as consuming a large amount of alcohol in a short period, is particularly damaging because it causes acute spikes in acetaldehyde and oxidative stress that completely overwhelm the liver's defense mechanisms. Furthermore, the modern alcohol landscape exacerbates these risks. The rise of high-gravity craft beers, sweet cocktails that mask the taste of ethanol, and the cultural normalization of wine o'clock have contributed to a creeping increase in daily consumption for many adults who would never identify as alcoholics. This functional heavy drinking is insidious because it sustains a constant level of hepatic inflammation without the dramatic social consequences that usually prompt intervention. We must also consider the synergistic toxicity of alcohol when combined with other risk factors. The investigative lens reveals that alcohol does not work in isolation. For individuals infected with hepatitis B or C, alcohol acts as a turbocharger for the virus, accelerating the progression to cirrhosis and cancer by decades. Similarly, the modern epidemic of obesity and metabolic syndrome has created a condition known as non-alcoholic fatty liver disease, NAFLD. When a person with an already fatty liver from poor diet engages in heavy drinking, the two conditions compound, creating a two-hit injury that destroys liver tissue faster than either factor could alone. Tobacco use, often paired with drinking, further multiplies the risk, as the carcinogens in smoke work in tandem with the solvent properties of alcohol to damage DNA. The societal implications of these are profound. We are facing a rising tide of alcohol-related liver disease in younger demographics. Historically seen as a disease of middle-aged men, alcoholic cirrhosis is now appearing in women in their 20s and 30s at alarming rates. Women are biologically more susceptible to alcohol-related liver damage due to lower levels of gastric ADH, meaning more alcohol reaches the liver intact, and a lower volume of body water, leading to higher concentrations of ethanol in the blood. The marketing machinery of the alcohol industry, which often targets women with wellness wines and low-calorie spirits, obscures this biological reality. Ultimately, the consumption of alcohol is a game of biological Russian roulette played with the body's most vital metabolic organ. The liver is incredibly resilient. It is the only organ capable of regenerating itself from a fraction of its original mass. However, this regenerative capacity is not infinite. Chronic heavy drinking exhausts the liver's ability to heal. The transition from fibrosis to cirrhosis to cancer is a one-way street, paved with collagen and mutated cells. By the time symptoms of liver cancer appear, jaundice, abdominal pain, unexplained weight loss, the disease is often advanced and incurable. The avoidance of heavy alcohol consumption is arguably the single most powerful preventative measure a person can take to preserve liver health. It requires a cultural shift, a recognition that the social lubricant is in fact a cellular abrasive. To lower the risk of liver cancer, one must respect the limitations of human biology and recognize that every drink exacts a price, one that the liver pays in silence until it can no longer afford the debt.
2. Moldy foods, moldy peanuts, corn, grains. In the shadowy corners of the global food supply chain, a microscopic assassin thrives. It is not a virus, nor a bacterium, but a fungus, specifically molds of the Aspergillus genus. While the modern consumer is conditioned to fear synthetic chemicals and artificial additives, one of the most potent naturally occurring carcinogens known to science is likely lurking in the pantry, invisible to the naked eye. This segment of the investigation centers on aflatoxins, a group of toxic compounds produced by Aspergillus flavus and Aspergillus parasiticus. These molds colonize staple crops such as corn, maize, peanuts, groundnuts, cottonseed, and tree nuts, turning the very foundation of human sustenance into a vehicle for liver cancer. The danger of aflatoxins lies in their extreme potency and their insidiousness. They are colorless, odorless, and tasteless in the quantities required to cause harm, yet they possess a terrifying specificity for destroying the genetic integrity of liver cells. The history of aflatoxin research reads like a medical thriller. The world was largely ignorant of this threat until 1960, when a mysterious plague struck the poultry industry in England. Within months, over 100,000 turkeys died from massive liver necrosis. The event, initially dubbed Turkey X disease, sparked an urgent scientific manhunt. Investigators eventually traced the cause to a shipment of peanut meal imported from Brazil that had been contaminated with a mold. This discovery unveiled the existence of aflatoxins and revolutionized our understanding of food safety. It revealed that the threat was not merely the mold itself, but the secondary metabolites, chemical weapons, that the mold produced under stress. Among the various types of aflatoxins identified, aflatoxin B1, AFB1, is the most toxic and the most prevalent. Its classification as a Group 1 carcinogen by the International Agency for Research on Cancer, IARC, places it in the highest tier of confirmed cancer-causing agents alongside asbestos and plutonium. The mechanism by which AFB1 induces liver cancer is a masterclass in molecular toxicology. When contaminated food is ingested, the aflatoxin is absorbed in the small intestine and transported to the liver. There it undergoes bioactivation. Ironically, it is the liver's attempt to detoxify the compound that renders it deadly.
Enzymes in the cytochrome P450 family, specifically CYP1A2 and CYP3A4, attempt to break down the aflatoxin, but in the process they convert it into a highly reactive intermediate called aflatoxin 8,9 epoxide. This epoxide is a chemical predator. It relentlessly seeks out DNA, specifically targeting the guanine bases within the genetic code. When it binds to DNA, it forms what is known as a DNA adduct, essentially a chemical barnacle that distorts the double helix. If the cell attempts to replicate with this adduct in place, errors occur. The most chilling aspect of aflatoxin toxicity is its precision. It has a distinct predilection for mutating the TP53 gene, often referred to as the guardian of the genome. The p53 protein produced by this gene is responsible for detecting DNA damage and ordering the cell to either repair itself or self-destruct, apoptosis. Aflatoxin induces a specific mutation at codon 249 of the TP53 gene. This mutation effectively disables the guardian. With the p53 protein neutralized, liver cells with damaged DNA are not killed off. Instead, they are allowed to survive and replicate, passing on their mutations to daughter cells. This loss of tumor suppression is a critical gatekeeper event that allows the uncontrolled cellular proliferation that defines cancer. The risk of aflatoxin exposure is inextricably linked to agriculture, climate, and economics. The Aspergillus fungus thrives in warm, humid environments. It infects crops not only in the field, particularly during drought stress, which weakens the plant's defenses, but also, and perhaps more dangerously, during post-harvest storage. If corn or peanuts are harvested while moist and then stored in silos or bags without adequate drying, the heat and humidity create an incubator for mold. This creates a stark geopolitical divide in liver cancer statistics. In the global north, including the United States and Europe, strict regulatory standards and advanced agricultural infrastructure minimize exposure. Crops are tested and contaminated batches are destroyed or relegated to non-food uses, though traces can remain. However, in the global south, particularly in sub-Saharan Africa, Southeast Asia, and parts of China, subsistence farming and a lack of industrial drying technology mean that millions of people are chronically exposed to high levels of aflatoxins in their daily diet. In these regions, hepatocellular carcinoma is a leading cause of cancer death,
directly tracking with the consumption of moldy maize and ground nuts. However, it would be a grave error to assume this is solely a third world problem. The globalized nature of the food trade means that imported nuts, spices, and grains can carry these toxins across borders. Furthermore, climate change is expanding the geographical range of Aspergillus. As summers become hotter and droughts more frequent in traditionally temperate zones, the aflatoxin belt is moving northward, threatening crops in regions that previously did not have to worry about this fungal contaminant. For the individual consumer, the danger is often disguised by the processed nature of food. You might not eat a visibly moldy peanut, but what about the peanut butter made from that nut? What about the corn chips made from contaminated maize flour? Aflatoxins are thermally stable. They are not destroyed by cooking, baking, or roasting. Once the toxin is in the grain, it remains there through the processing chain. This is why natural or organic peanut butters, which may lack the preservatives that inhibit mold growth or bypass the rigorous testing of major industrial brands, can paradoxically pose as higher risk if not sourced from strictly monitored supply chains. The visual identification of danger is also tricky. While the advice to avoid moldy foods seems straightforward, the mold that produces aflatoxin is not always the fuzzy green growth one sees on old bread. Aspergillus can penetrate deep into the kernel of corn or the meat of the nut without leaving obvious external signs. Moreover, there is a dangerous misconception that one can simply cut off the moldy part of a food item and eat the rest. This is a gamble with lethal stakes. The visible mold is merely the reproductive structure of the fungus. The mycelium, the root structure, extends deep into the food, invisible to the eye, carrying the toxin with it. In porous foods like breads, grains, and nuts, if you see mold, the entire entire biological system of that foodstuff is likely compromised. The investigation into aflatoxins also reveals a terrifying synergy with hepatitis B. This is a prime example of biological cooperation between killers. In regions where both hepatitis B prevalence and aflatoxin exposure are high, such as parts of China and West Africa, the rate of liver cancer is vastly higher than the sum of the two risks individually.
They work via different mechanisms. The virus causes chronic inflammation and cell turnover, while the toxin provides the specific genetic mutation to accelerate the path to cancer. An individual with chronic hepatitis B who consumes aflatoxin-contaminated food faces a risk of liver cancer that is up to 60 times higher than someone with neither risk factor. To lower the risk of liver cancer, a zero-tolerance policy for mold on grains and nuts is essential. This extends beyond merely throwing away a bad bag of peanuts. It involves a conscious curation of one's diet. It means storing nuts, grains, and seeds in cool, dry places, or freezing them to inhibit fungal growth. It means being wary of budget bulk bins in stores with low turnover, where humidity can fluctuate. It involves understanding that dry foods are not dead. They are dormant biological entities susceptible to infection. The narrative of moldy food is a reminder of humanity's fragile relationship with nature. We compete with fungi for the same energy sources. When Aspergillus colonizes a pea mote, it is staking a claim. By producing aflatoxin, it is defending that claim. When we ingest that toxin, we become collateral damage in an ancient biological turf war. The liver, in its attempt to process this complex organic poison, falls victim to a chemical sabotage so precise that it rewrites the genetic code of life itself. Avoiding these foods is not just a matter of hygiene. It is a tactical maneuver to deny entry to a molecular saboteur that has claimed millions of lives through the silent, invisible corruption of our daily bread.
3. Processed meats: sausage, bacon, deli meat. To enter the world of processed meats is to step into a triumph of industrial chemistry over biological narrative. It is a realm where the concept of freshness is suspended in time, preserved not by nature, but by a potent cocktail of salts, nitrates, and smoke. We walk through the aisles of the supermarket, seduced by the vibrant pink hues of ham, the marbled perfection of bacon, and the uniform geometry of sausages. Yet this visual appeal is a carefully constructed illusion, a masquerade that hides one of the most significant dietary drivers of carcinogenesis in the modern world. In 2015, the International Agency for Research on Cancer, IARC, the specialized cancer agency of the World Health Organization, dropped a bombshell that shook the global food industry to its core. Processed meats were classified as Group 1 carcinogens. This designation placed hot dogs, salami, and pepperoni in the same hazard category as tobacco smoke, asbestos, and plutonium. While the potency differs, eating a slice of bacon is not statistically equivalent to smoking a pack of cigarettes. The level of evidence regarding causality is identical. We know with absolute scientific certainty that these foods cause cancer. While much of the public discourse focused on colorectal cancer, the implications for the liver are equally profound, insidious, and chemically distinct. The definition of processed meat covers any meat that has been modified to extend its shelf life or change its taste. This includes curing, smoking, salting, or the addition of chemical preservatives. The primary villain in this narrative is the nitrogen atom, specifically in the form of sodium nitrite, NaNO2, and sodium nitrate, NaNO3. These compounds are the magic dust of the meat industry. Without them, bacon would turn a repulsive shade of gray-brown, and botulism, a deadly neurotoxic illness, would be a constant threat in cured sausages. Nitrites bind to myoglobin, the iron-containing protein in muscle, to form nitrosomyoglobin, which gives cured meats their characteristic, appetizing pink color. They also infuse the meat with a sharp, piquant flavor that the human palate craves. However, when these nitrates and nitrites enter the human body, the preservative becomes a precursor. The liver is the central processing unit for everything we ingest, and it is here that the nitrate paradox unfolds.
When you consume a pepperoni pizza or a breakfast sausage, the nitrates are absorbed and enter the bloodstream. Under specific conditions, such as the high heat of cooking or the acidic environment of the stomach, nitrites react with amines, compounds derived from the breakdown of proteins, to form N-nitroso compounds, specifically nitrosamines. Nitrosamines are among the most potent hepatotoxins, liver toxins, known to toxicology. In fact, one specific nitrosamine found in processed meats, N-nitrosodimethylamine, NDMA, is so effective at destroying liver cells that it is used by research scientists to deliberately induce liver fibrosis and liver cancer in laboratory rats for drug testing. When we consume processed meats, we are essentially microdosing ourselves with the very agent used to model liver disease in the lab. The mechanism of nitrosamine-induced liver cancer is a violent molecular assault. The liver attempts to metabolize these compounds using the cytochrome P450 enzyme system, specifically the CYP2E1 isozyme. This metabolic process, intended to render the toxin harmless, inadvertently activates it. The metabolism of nitrosamines produces highly reactive alkylating agents. These agents are chemical saboteurs that attack the DNA within the nucleus of the liver cell, hepatocyte.
They add alkyl groups to the guanine bases of the DNA, causing structural instability. If the DNA repair mechanisms of the cell fail to correct this damage before the cell divides, the mutation becomes permanent. Over decades of breakfast buffets and ballpark franks, these mutations accumulate, eventually silencing tumor suppressor genes or activating oncogenes, leading to the uncontrolled cell growth that defines hepatocellular carcinoma, HCC. Furthermore, the danger of processed meat extends beyond nitrates. We must consider the heme iron factor. Red meat, the base for most processed products, is rich in heme iron. While iron is essential for life, free iron is a pro-oxidant. In the context of processed meats, the heme iron acts as a catalyst for the endogenous formation of N-nitroso compounds within the gut. Even if the manufacturer reduces the added nitrites, the heme iron in the meat can interact with other nitrogen sources in the diet to create carcinogens internally. The liver, which stores the body's iron reserves, is particularly susceptible to iron-induced oxidative stress. Excess iron accumulation in the liver creates a state of chronic inflammation,
acting as fuel for the fire started by the nitrosamines. Then there is the issue of sodium. Processed meats are essentially salt delivery vehicles. A few strips of bacon or a serving of deli turkey can contain a staggering percentage of the daily recommended sodium intake. This massive influx of salt forces the body to retain water and increases blood pressure, altering the hemodynamics of the liver. But more critically, high sodium intake is linked to cellular hypertrophy and fibrosis. In a liver that is already struggling with the metabolic load of nitrates and lipids, the osmotic stress caused by excess sodium weakens the cellular integrity, making the hepatocytes more vulnerable to carcinogenic attack. The industrialization of meat processing has also introduced a philosophical shift in how we view food. We have moved from whole food to food product. Mechanically separated meat, the slurry of muscle, fat, and connective tissue blasted off the bone by high-pressure water, is the base for many sausages and nuggets. This slurry is a blank canvas that requires heavy chemical intervention to be palatable. Phosphates are added to bind water, selling you expensive water weight and improve texture. Hydrolyzed vegetable proteins are added to boost the umami flavor. The result is an ultra-processed matrix of fats and additives that promotes obesity and insulin resistance. Non-alcoholic fatty liver disease, NAFLD, is now the fastest-growing cause of liver cancer in the West, and processed meats are a primary dietary driver of this epidemic. The high saturated fat content, combined with the inflammatory additives, encourages the liver to store fat. A fatty liver is an inflamed liver, and inflamed liver is a precancerous liver. The uncured deception deserves specific scrutiny. In response to consumer fear of nitrates, the industry has flooded the market with natural or uncured bacon and hot dogs. The labels proudly proclaim no nitrates or nitrites added. However, look closely at the asterisk, except for those naturally occurring in celery powder. This is a masterpiece of chemical semantics. Celery is naturally high in nitrates. Manufacturers concentrate celery juice, convert the nitrate to nitrite using bacterial cultures, and add this powder to the meat. Chemically, the nitrate molecule derived from celery powder is identical to the nitrate molecule in the pink synthetic curing salt. The liver cannot tell the difference.
It processes them exactly the same way, creating the same carcinogenic nitrosamines. The natural label is a regulatory loophole, not a safety shield. It allows consumers to feel virtuous while consuming the same class of carcinogens. The cultural entrenchment of processed meat makes this entry particularly difficult to navigate. These foods are woven into our nostalgia and our national identities. The German bratwurst, the Italian prosciutto, the American hot dog, the British bacon buddy, these are culinary pillars. Yet the epidemiological data is unyielding. Studies tracking hundreds of thousands of people over decades have consistently shown that high consumption of processed meat is associated with increased mortality from all causes, including liver disease. The risk is dose-dependent. There is no clear threshold below which the risk is zero, though occasional consumption poses a lower threat than daily habit. We are also witnessing a disturbing trend in the formulation of these products with the addition of smoke flavor. While traditional smoking involves exposing meat to actual wood smoke, which carries its own risks detailed in the next entry, liquid smoke and chemical flavorings add another layer of complexity for the liver to detoxify. These synthetic compounds are often proprietary mixtures, the long-term hepatic effects of which are not fully mapped. We are essentially conducting a massive uncontrolled toxicology experiment on the human population. To lower liver cancer risk, the elimination or drastic reduction of processed meats is a non-negotiable directive. It requires a rejection of the convenience and hyperpalatability engineered by food scientists. It means returning to fresh, intact cuts of meat that have not been injected, cured, or ground into a paste with preservatives. It means understanding that the vibrant pink color of a ham sandwich is a chemical warning sign, a flag signaling the presence of compounds that possess the power to rewrite the genetic code of your liver cells. The investigation into processed meat reveals a stark truth. In our quest to conquer the decay of flesh, to make meat last forever on the shelf, we have inadvertently shortened the shelf life of the human drinker.
4. Charred or burnt meat, BBQ burnt parts. Fire is the element that forged humanity. The discovery that cooking meat makes it easier to digest and releases more calories was a pivotal moment in our evolutionary history, allowing for the rapid expansion of the human brain. We are the cooking ape. However, this ancient alliance with fire contains a dark paradox. The very chemical reaction that makes grilled meat delicious, the searing, the browning, the char, is also the reaction that generates potent mutagens. When we stand around the barbecue, mesmerized by the flames licking a steak or the smoke rising from a rack of ribs, we are witnessing the genesis of environmental carcinogens. The entry regarding charred and burnt meat is not about an additive or a preservative. It is about the fundamental physics of heat and organic matter. It is about what happens when muscle tissue meets extreme temperatures, creating a chemical profile that the liver is ill-equipped to handle. The investigation centers on two distinct classes of carcinogenic compounds formed during high-temperature cooking: heterocyclic amines, HCAs, and polycyclic aromatic hydrocarbons, PAHs. These are not present in the raw meat; they are alchemized by the cooking process. Let us first dissect heterocyclic amines, HCAs. These compounds are the product of a collision between amino acids, the building blocks of protein, sugars, and creatine. Creatine is a substance found in muscle tissue that provides energy for contraction. When meat, be it beef, pork, fish, or poultry, is subjected to temperatures exceeding 300 degrees Fahrenheit, 150 degrees Celsius, and particularly when it is cooked for a long duration, these molecules react chaotically. The result is the formation of HCAs, such as PHIP and MyLQX. These acronyms represent complex ring structures that are highly mutagenic. It is crucial to understand that this is not just about red meat. In fact, grilled chicken and fish can produce even higher concentrations of HCAs than beef because they often contain higher levels of specific amino acids and creatine. The formation of HCAs is directly proportional to the temperature and the level of doneness. The blackened, crispy crust that many gourmands prize is essentially a concentrated deposit of these toxins. When you ingest that piece of well-done charred steak, the HCAs are absorbed and transported to the liver. The liver, recognizing them as xenobiotics, foreign biological substances,
initiates phase 1 metabolism to excrete them. The primary enzyme responsible for this is CYP1A2. In a cruel twist of metabolic fate, the liver processes HCAs into an intermediate form, N-hydroxy-HCAs, that is even more toxic than the original compound. This is bioactivation. These activated metabolites can travel through the bloodstream or remain in the liver, where they bind covalently to DNA. They form bulky adducts that distort the DNA helix. When the liver cells attempt to replicate, the replication machinery trips over these adducts, inserting the wrong genetic base or causing a break in the DNA strand. This is the spark of mutagenesis that can lead to hepatocellular carcinoma. The second class of villains, polycyclic aromatic hydrocarbons, PAHs, forms through a different mechanism. PAHs are not born inside the meat, they are born in the fire. When fat and juices from the meat drip down onto the hot coals, wood, or heating element, they undergo pyrolysis, thermal decomposition. This creates smoke. The smoke is rich in PAHs, such as benzo-A-pyrene, the same carcinogen found in cigarette smoke and coal tar. As the smoke rises, it envelops the meat, depositing these lipophilic, fat-loving chemicals onto the surface. If you are eating smoked brisket or a burger flared up by grease fires, you are eating a sponge saturated with PAHs. Unlike HCAs, which are formed deep within the muscle fibers, PAHs coat the exterior. The liver's struggle with PAHs is exhaustive. These are large, complex molecules that are difficult to break down. The detoxification process generates massive amounts of reactive oxygen species, ROS, contributing to the oxidative stress burden of the liver. This oxidative stress damages the lipid membranes of liver cells and depletes the organ's antioxidant reserves. For a liver that may already be compromised by alcohol or fatty diet, the additional burden of detoxifying a weekend's worth of barbecue carcinogens can be the straw that breaks the camel's back. There is a significant genetic component to this risk, adding a layer of unpredictability. The enzymes that metabolize these toxins, particularly N-acetyltransferase, NAT2, vary wildly between individuals. People are categorized genetically as rapid or slow acetylators. In the context of HCAs, rapid acetylators may activate the toxins more quickly, while slow acetylators may allow them to linger. This genetic lottery means that two people can eat the same burnt burger,
and one may suffer significantly more DNA damage than the other. Because we do not routinely test for these enzyme phenotypes, every meal of charred meat is a gamble with one's personal genetic susceptibility. The cultural resistance to this information is immense. Grilling is more than a cooking method. It is a ritual of masculinity, summer, and celebration. The Maillard reaction, the chemical process that browns food and creates complex flavors, is the holy grail of the culinary world. Chefs are trained to seek the sear, to build the crust. To suggest that this crust is carcinogenic is to attack the very foundation of flavor. However, the chemistry does not care about culinary tradition. The darker the meat, the higher the concentration of mutagens. Studies have shown that very well-done meat can contain HCA concentrations thousands of times higher than meat cooked to medium rare or prepared via boiling or stewing.
The investigation must also touch upon the definition of burnt. It is not just the black charcoal-like bits that are dangerous. Even deep browning indicates the presence of these compounds. The misconception is that if you scrape off the black part, you are safe. While this reduces the load of PAHs, which are on the surface, the HCAs are formed throughout the meat where the heat has penetrated. You cannot scrape away a chemical reaction that has occurred at the molecular level of the muscle fiber. Are there ways to mitigate this without abandoning the grill entirely? Science offers some nuanced solutions, though they require a change in behavior. Marinating meat before grilling has been shown to drastically reduce HCA formation. Marinades containing herbs rich in antioxidants like rosemary, thyme, and garlic, or acidic ingredients, lemon juice, vinegar, act as a chemical barrier, preventing the formation of the mutagenic rings even under high heat. Flipping the meat frequently prevents the surface temperature from staying high enough for long enough to generate massive amounts of toxins. Pre-cooking meat in a microwave or oven and then finishing it on the grill for flavor reduces the time of exposure to extreme heat. However, the safest path for the liver remains the avoidance of direct flame and char. We must also consider the future of cooking technology in this context. The rise of sous vide cooking, water bath cooking at precise
low temperatures represents a technological evolution that prioritizes safety and texture over the primal char. By cooking a steak at 130 degrees Fahrenheit for 2 hours, one achieves perfect tenderness without ever reaching the temperature threshold for HCA formation. The liver is spared the assault. As our understanding of food chemistry advances, the burnt ends of the barbecue world may eventually be viewed with the same wariness as we now view lead pipes or unfiltered cigarettes, relics of the time when we enjoyed the utility of a thing without understanding its toxicity. Ultimately, the consumption of charred and burnt meat is a direct injection of mutagenic chaos into the hepatic system. The liver is a resilient organ, capable of immense feats of detoxification, but it was not evolutionarily designed to handle the concentrated chemical byproducts of pyrolysis on a regular basis. Every bite of charred flesh requires the liver to engage in a high-stakes game of molecular disposal, where a single error in processing can result in a permanent mutation. To lower the risk of liver cancer, one must tame the fire. We must learn to appreciate the flavors of food, not for how violently they have been burned, but for how gently they have been prepared. The flavor of fire is, biologically speaking, the flavor of damage.
5. Deep-fried foods, fried chicken, fries. The allure of deep-fried food is an evolutionary trap. Humans are biologically hardwired to seek out energy-dense sustenance, and there is no more efficient delivery system for calories than food submerged in boiling fat. The sensory experience of biting into a piece of fried chicken or a French fry, the audible crunch of the crust, the immediate release of salt, and the mouth-coating richness of lipids triggers a dopamine response in the brain that rivals the effects of narcotics. However, this culinary technique, which essentially dehydrates food while simultaneously forcing oil into its cellular structure, represents a catastrophic assault on the liver. The danger of deep-fried foods extends far beyond the simplistic metric of calories in, calories out. It is a matter of complex lipid chemistry, thermal degradation, and the ingestion of compounds that act as direct inflammatory agents within hepatic tissue. To understand why fried foods are a primary driver of liver cancer risk, we must investigate the molecular violence that occurs inside the deep fryer. The primary medium of this assault is the oil itself. Historically, humans cooked with stable, saturated animal fats like lard, tallow, or butter, which are relatively resistant to heat. The 20th century, however, ushered in the era of industrial vegetable oils, soybean, corn, canola, and cottonseed. These oils are predominantly polyunsaturated fats, PUFAs. While often marketed as heart-healthy, PUFAs are chemically unstable due to their multiple double bonds. When these oils are subjected to the extreme temperatures of a deep fryer, typically between 350 degrees Fahrenheit and 375 degrees Fahrenheit, and particularly when they are exposed to oxygen and moisture from the food, they undergo a rapid and violent degradation process known as peroxidation. This thermal stress causes the oil molecules to fracture and recombine into new toxic structures. One of the most dangerous byproducts of this process is a class of compounds called aldehydes. Specifically, the heating of vegetable oils generates 4-hydroxynonenal, 4-HNE, a highly toxic alpha-beta unsaturated aldehyde. In the scientific community, 4-HNE is often referred to as a marker of oxidative stress, but it is more accurately described as an agent of cellular destruction. When you consume deep-fried foods, you are ingesting significant quantities of 4-HNE. Upon reaching the liver, this compound attacks functional proteins, DNA,
and phospholipids. It interferes with cell signaling and promotes mitochondrial dysfunction. Research has shown that 4-HNE is intimately involved in the pathogenesis of alcoholic and non-alcoholic liver diseases, acting as a potent driver of inflammation and fibrosis. It essentially cooks the liver cells from the inside out, creating a toxic environment where DNA repair mechanisms are suppressed and mutation becomes more likely. The peril is compounded by the commercial reality of the restaurant industry. In a home kitchen, oil might be used once or twice. In a fast food establishment or a casual dining restaurant, the economics dictate that oil must be used for days, sometimes upwards of a week before being discarded. With every hour of heating, the oil degrades further, the smoke point lowers, and the concentration of polar compounds, breakdown products that have been linked to hypertension and organ damage, skyrockets. This abused oil becomes a viscous sludge of polymers and cyclic monomers. When you order fries from a place that hasn't changed its oil recently, you are not just eating potatoes and oil, you are consuming a concentrated chemical slurry of thermal degradation products. These compounds place an immense detoxification load on the liver, depleting its stores of glutathione, the master antioxidant, and leaving the organ vulnerable to oxidative injury. We must also address the specter of trans fats. While legislative bans in the United States and Europe have largely removed artificial trans fats, partially hydrogenated oils, from the supply chain, the chemistry of frying can create thermal trans fats. When unsaturated oils are heated to high temperatures for long periods, some of the cis bonds in the fat molecules can flip to a trans configuration. Furthermore, many restaurants still use frying blends that are high in omega-6 fatty acids, which are pro-inflammatory. The modern diet is already drowning in omega-6s relative to omega-3s, creating a state of chronic low-grade inflammation throughout the body. The liver is the epicenter of this inflammation. The influx of oxidized omega-6 fatty acids from fried foods activate Kupffer cells, the resident macrophages of the liver. Once activated, these cells release cytokines like tumor necrosis factor alpha, TNF-alpha, and interleukin-6, IL-6, which signal the liver tissue to become inflamed and eventually to scar.
The connection between deep-fried foods and hepatocellular carcinoma, HCC, is mediated largely through the gateway disease of non-alcoholic fatty liver disease, NAFLD, and its more severe form, non-alcoholic steatohepatitis, NASH. Fried foods are calorically dense and hyperpalatable, leading to overconsumption and visceral obesity. However, the specific type of fat matters. Oxidized lipids from fried foods are preferentially stored in the liver rather than in subcutaneous fat tissue. This leads to hepatic steatosis, fatty liver. But unlike the bland fat storage of simple obesity, the fat stored from fried foods is biologically active and toxic, lipotoxicity. It induces stress in the endoplasmic reticulum of the hepatocytes. As the liver cells become overwhelmed by lipid peroxidation, they begin to die, apoptosis. The liver attempts to repair this damage by laying down collagen fibers, leading to fibrosis and cirrhosis. As we have established, cirrhosis is the fertile soil in which liver cancer grows. Additionally, the frying of starchy foods like potatoes and breading creates another carcinogen, acrylamide. Discovered in food only in 2002, acrylamide forms via the Maillard reaction when the amino acid asparagine reacts with reducing sugars at high temperatures. While the primary concern with acrylamide has historically been neurological damage and reproductive toxicity, animal studies have consistently shown it to be a multi-organ carcinogen. The liver, as the metabolic clearinghouse for acrylamide via the cytochrome P452E1 enzyme, converts it into glycidamide, a highly reactive epoxide that binds to DNA and causes mutations. While the epidemiological evidence in humans is still evolving, the biological plausibility of acrylamide contributing to the cumulative carcinogenic load on the liver is high, particularly in heavy consumers of French fries and potato chips. The cultural dimension of deep-fried food is a testament to the industrialization of the global palate. The southern fried tradition in the United States, originally a special occasion method of preparation due to the labor and cost of lard, has been transformed by industrial agriculture into a daily dietary staple. In many socioeconomically disadvantaged areas, often termed food deserts, deep-fried fast food is the most accessible and affordable source of calories. This creates a terrifying class disparity in liver cancer risk.
We're seeing a dramatic rise in NASH-related hepatocellular carcinoma in populations that do not drink alcohol but consume high amounts of industrial fried foods. The Western diet, characterized by high intake of fried and processed foods, is being exported globally, bringing with it a tsunami of metabolic liver disease to countries like China, India, and Mexico, where liver cancer rates are already a significant concern due to viral hepatitis. Furthermore, the breading on fried foods acts as a sponge, soaking up the oxidized oil. A piece of fried chicken can consist of up to 40% fat by weight, most of it residing in the crust. This breading is often highly processed wheat flour, a refined carbohydrate that spikes blood sugar and insulin. When you combine the oxidized fats with the insulin spike, you create the perfect metabolic storm for fat deposition in the liver. High insulin levels lock the fat in the liver, preventing it from being burned for energy. This combination, refined carb plus oxidized fat, is the hallmark of the modern obesogenic environment. To lower the risk of liver cancer, one must view deep-fried foods not as a treat, but as a source of environmental toxins.
The occasional homemade item fried in fresh, high-quality oil, like avocado oil or tallow, poses a negligible risk compared to the habitual consumption of industrial fast food. However, the goal should be a drastic reduction. The liver possesses a remarkable ability to heal, and removing the constant bombardment of aldehydes and lipid peroxides allows the organ to reduce inflammation and clear stored fat. But as long as the deep fryer remains the engine of the modern diet, the liver will remain under siege, fighting a chemical war it eventually loses to the chaotic proliferation of cancer cells.
6. Sugary drinks, soda, sweet tea. If alcohol is the overt assassin of the liver, sugary drinks are the silent strangler. They are the single most significant dietary contributor to the obesity epidemic and the exploding rates of metabolic liver disease worldwide. When we investigate the etiology of liver cancer in the 21st century, we must look beyond the bar tap and toward the soda fountain. The consumption of liquid sugar, specifically in the form of sugar-sweetened beverages, SSBs, like soda, sweet tea, energy drinks, and fruit juices, represents a unique and devastating challenge to hepatic physiology. The danger lies not just in the calories, but in the specific molecule of fructose and the speed at which it is delivered to the system. Unlike glucose, which is a fuel source utilized by every cell in the human body, fructose is a hepatotoxin in high doses. It is metabolized almost exclusively by the liver. In this way, the metabolic burden of a soda is remarkably similar to that of a beer. To understand this, we must dissect the composition of modern sweeteners. High fructose corn syrup, HFCS, and sucrose, table sugar, are roughly 50% glucose and 50% fructose. When you drink a 20-ounce soda, you are ingesting approximately 65 grams of added sugar. In liquid form, there is no fiber to slow absorption. The gut is flooded. The glucose spikes your blood sugar, triggering the pancreas to release insulin. The fructose, however, bypasses this insulin regulation and goes straight to the liver via the portal vein. This is where the catastrophe begins. The liver is suddenly inundated with fructose, far more than it can use for energy production. It has no choice but to activate a metabolic pathway called de novo lipogenesis, DNL. DNL is the process of turning sugar into fat. The liver converts the excess fructose into triglycerides, fat molecules. Some of these triglycerides are exported into the bloodstream as VLDL cholesterol, contributing to heart disease, but a significant portion remains trapped within the liver cells. This is the genesis of non-alcoholic fatty liver disease, NAFLD. It is estimated that a daily habit of sugary drinks can induce fatty liver changes in as little as 6 months. The liver becomes marbled with fat, much like a piece of foie gras, which is notably produced by force-feeding ducks corn, a high-carbohydrate grain. But the damage caused by liquid fructose goes deeper than fat storage.
The metabolism of fructose in the liver consumes adenosine triphosphate, the cellular energy currency. This rapid depletion of ATP induces a state of energy shock in the liver cells. As the cells scramble to recover, the breakdown of ATP generates uric acid as a waste product. Uric acid is typically associated with gout, but within the liver it acts as a pro-inflammatory signal. It induces oxidative stress within the mitochondria, the power plants of the cell. This mitochondrial dysfunction impairs the liver's ability to burn fat, creating a vicious cycle. The liver makes more fat from the sugar, but loses the ability to burn that fat due to the mitochondrial damage caused by the sugar metabolism. The state of toxic fat accumulation leads to insulin resistance. The liver, choked with fat and inflamed by uric acid, stops responding to insulin signals. The pancreas compensates by pumping out even more insulin. This results in hyperinsulinemia, chronically high levels of insulin in the blood. Here's the critical link to cancer. Insulin is a potent growth factor. It is anabolic. It tells cells to grow and divide. High levels of insulin increase the bioavailability of insulin-like growth factor 1, a hormone that has been strongly linked to the development of various cancers, including hepatocellular carcinoma. In an environment of chronic inflammation and high insulin, precancerous liver cells are given the green light to proliferate. The cultural context of sugary drinks is one of aggressive normalization. In the American South, sweet tea is not just a beverage, it is a cultural institution. It is often supersaturated with sugar, containing concentrations that rival or exceed soda. Similarly, the big gulp culture of 32-ounce and 64-ounce sodas has normalized serving sizes that would have been considered lethal doses of sugar a century ago. We have raised entire generations on liquid sugar, habituating their palates and their metabolic set points to a level of sweetness that does not exist in nature. The marketing of these drinks, often tying them to happiness, youth, and vitality, masks the reality that they are metabolic poisons. The big soda industry has borrowed the playbook of the tobacco industry, funding studies to cast doubt on the link between sugar and disease and lobbying against taxes and regulations. We must also scrutinize the role of fruit juice, often perceived as a healthy alternative.
Fruit juice is essentially soda with vitamins. When you juice an orange or an apple, you strip away the fiber, the antidote to the poison. The fiber in whole fruit creates a gelatinous matrix in the gut that slows the absorption of sugar, allowing the liver to process it slowly. Without the fiber, the fructose hits the liver with the same violent velocity as a cola. Studies have shown that high consumption of fruit juice is associated with an increased risk of type 2 diabetes and fatty liver, debunking the health halo that surrounds these products. The progression from sugary drink consumption to liver cancer is a slow, silent burn. It starts with a teenager drinking 2 sodas a day. By their 20s, they have early-stage fatty liver, steatosis, often asymptomatic. By their 30s, the constant inflammation from lipotoxicity and uric acid leads to non-alcoholic steatohepatitis, NASH. Scar tissue begins to form, fibrosis. By their 50s, they may have cirrhosis, all without ever having been a heavy drinker of alcohol. Once cirrhosis sets in, the genomic instability skyrockets and the path to cancer is paved. This is the new face of liver cancer, a patient who never touched a drop of whiskey but drank a liter of cola every day for 3 decades. Furthermore, the interaction between fructose and the gut microbiome is an emerging field of horror. High sugar intake alters the bacterial population in the intestines, promoting the growth of pathogenic bacteria. These bacteria produce endotoxins, lipopolysaccharides, that can leak through the gut wall, a condition known as leaky gut. These endotoxins travel directly to the liver via the portal vein, triggering a massive immune response. This gut-liver axis of inflammation is a powerful promoter of liver tumorigenesis. The sugar feeds the bad bacteria poison the gut lining, and the liver takes the hit. The economic implications are staggering. The cost of treating NAFLD and its complications is threatening to bankrupt healthcare systems. Liver transplants, once primarily the domain of hepatitis C and alcoholic cirrhosis patients, are increasingly being performed for NASH. But transplantation is a desperate end-stage solution. The prevention lies in the removal of the vector. To lower liver cancer risk, the elimination of sugary drinks is arguably the most effective dietary intervention available. It is a binary choice: drink water, unsweetened tea, or black coffee.
And the liver's metabolic burden drops instantly. The liver can clear the accumulated fat if the influx of fructose stops. Insulin sensitivity can be restored, but as long as the liquid sugar flows, the liver remains a factory for its own destruction. The sweet taste on the tongue is a deception. Biologically, it is a signal of impending metabolic collapse. In the hierarchy of carcinogenic foods, sugary drinks occupy a unique position. They do not contain a carcinogen per se, but they create the physiological environment: obesity, diabetes, inflammation, cirrhosis, that makes cancer inevitable. They are the terraformers of the body, reshaping the internal landscape into a hospitable home for malignancy.
7. Ultra-processed snacks, chips, packaged cakes. To walk down the center aisles of a modern supermarket is to traverse a landscape of chemical engineering disguised as food. Here, in the brightly colored rows of potato chips, cheese puffs, snack cakes, and crackers, we encounter the apex of the ultra-processed phenomenon. These are not merely foods that have been cooked, they are industrial formulations, deconstructed from their original botanical sources and reconstructed using sophisticated technology to achieve a level of hyperpalatability that overrides the body's satiety signals. While the public often views these items as harmless indulgences or mere sources of empty calories, the investigative lens reveals a far more sinister reality. Ultra-processed snacks are metabolic disruptors that place a unique and heavy toxicological burden on the liver. They are the delivery vehicles for a host of synthetic additives, emulsifiers, and packaging contaminants that collectively degrade hepatic function and pave the road toward hepatocellular carcinoma. The definition of ultra-processed comes from the NOVA classification system, a framework developed by researchers at the University of Sao Paulo to categorize food not by nutrient content, but by the extent and purpose of industrial processing. Group 4, the ultra-processed category, consists of formulations made mostly or entirely from substances derived from foods and additives with little, if any, intact whole food. When you consume a bag of flavored tortilla chips or a packaged snack cake, you are essentially eating a food matrix that has been pre-digested by machines. The fiber has been pulverized or removed, the protein denatured, and the starches modified. This physical degradation means that the glucose and fat contained within these snacks hit the bloodstream with violent speed. The liver, which acts as the metabolic gatekeeper, is not designed to handle this rapid influx of substrates. It is akin to trying to drink from a fire hose. The overwhelming volume and velocity of energy force the liver into a state of panic, triggering immediate lipogenesis, the conversion of this excess energy into visceral fat. However, the caloric density is only the surface of the problem. The true danger of ultra-processed snacks lies in the cosmetic chemicals used to make them durable, attractive, and addictive. Let us examine the role of emulsifiers such as carbomethylcellulose, CMC, and polysorbate 80.
These compounds are ubiquitous in packaged cakes and creamy snacks, used to improve texture and extend shelf life. Recent research has uncovered a terrifying link between these additives and liver health via the gut-liver axis. The human gut is lined with a protective mucus layer that separates the microbiome from the intestinal wall. Emulsifiers act like detergents in the gut. They strip away this mucus layer, allowing bacteria to come into direct contact with the intestinal cells. This induces leaky gut, or increased intestinal permeability. When the gut barrier is breached, bacterial byproducts, specifically lipopolysaccharides, or LPS, also known as endotoxins, leak into the bloodstream. Because the blood from the gut drains directly into the portal vein, the liver is the first organ to be bathed in these toxins. The arrival of LPS in the liver triggers a massive immune response. The Kupffer cells, the liver's resident macrophages, detect the endotoxins and release a storm of inflammatory cytokines, including tumor necrosis factor alpha. This state of chronic low-grade inflammation, driven by the daily consumption of emulsifiers and snack foods, is a potent promoter of liver fibrosis and cancer. The liver is essentially fighting a perpetual infection caused not by a virus, but by the ingredients in a snack cake.
Furthermore, we must investigate the preservatives used to keep these snacks fresh for months or years. Compounds like butylated hydroxyanisole, or BHA, and butylated hydroxytoluene, or BHT, are commonly sprayed onto the packaging liners or added directly to the fats in chips and crackers to prevent rancidity. The National Toxicology Program has classified BHA as reasonably anticipated to be a human carcinogen. While the FDA considers them safe in limited quantities, these are cumulative toxins. The liver is responsible for metabolizing these synthetic phenols. In animal studies, high doses of BHA and BHT have been shown to induce liver enlargement and promote the development of liver tumors. By consuming these snacks daily, we are conducting a long-term, uncontrolled toxicology experiment on our own bodies, testing the limits of the liver's detoxification pathways. The investigation into ultra-processed snacks must also address the packaging paradox. The food itself is often secondary to the vessel that contains it. Snack bags, wrappers, and trays are sophisticated feats of material science.
Often coated with per- and polyfluoroalkyl substances, PFAS, the so-called forever chemicals, to make them grease-resistant. If you have ever wondered why the oil from a bag of chips doesn't seep through the paper, the answer is often PFAS. These chemicals are notorious for their persistence. They do not break down in the environment or the human body. When they migrate from the packaging into the food, a process accelerated by heat and the fatty nature of the snacks, they are ingested and accumulate in the liver. PFAS have been structurally linked to liver damage, including altered lipid metabolism and immune system suppression. The Environmental Protection Agency, EPA, has increasingly flagged these compounds as hazardous, yet they remain prevalent in the fast-moving consumer goods sector. Thus, the snack serves as a Trojan horse, smuggling environmental pollutants directly into the hepatic system. We must also consider the nutrient displacement theory. The liver is a nutrient-hungry organ. To perform its Phase 1 and Phase 2 detoxification processes, the biochemical assembly lines that neutralize toxins, it requires specific micronutrients, methylation factors like folate, B12, and choline, antioxidants like vitamin C and E, and sulfur-containing amino acids. Ultra-processed snacks are chemically engineered to be empty. They provide fuel, calories, without the machinery, nutrients, required to burn it. When a diet is dominated by chips and cakes, the liver is starved of the very tools it needs to protect itself. A choline-deficient diet, for example, is a well-established method for inducing fatty liver and liver cancer in laboratory animals. By replacing nutrient-dense whole foods with ultra-processed substitutes, we are systematically dismantling the liver's defense systems, leaving it vulnerable to DNA damage. The psychology of the snack industry reveals a calculated effort to bypass biological control mechanisms. Food scientists utilize the bliss point, the precise ratio of salt, sugar, and fat that maximizes sensory pleasure and overrides the stop signal in the brain. This is not accidental, it is predatory. The texture is engineered to have a high vanishing caloric density. Think of how a cheese puff melts in your mouth. This tricks the brain into thinking you haven't eaten anything substantial, encouraging continuous consumption. This leads to a state of chronic hyperfeeding and obesity,
which is the strongest driver of non-alcoholic steatohepatitis, NASH. NASH is quickly becoming the leading cause of liver transplantation and liver cancer in the Western world. The causality is clear. The engineered addiction to ultra-processed snacks leads to metabolic syndrome, which leads to a fatty, inflamed, and eventually cancerous liver. Moreover, the ingredients list of these products often hide sources of fructose under obscure names. Modified cornstarch, tapioca syrup, and crystalline fructose are all ways to introduce rapid-acting carbohydrates that spike hepatic lipogenesis. The gluten-free trend has ironically exacerbated this issue in the snack world, as manufacturers often replace wheat flour with highly refined starches, potato, rice, tapioca, that have an even higher glycemic index and lack the protein structure of wheat. The liver, forced to process these refined starches, is pushed further into a fat-storing mode. The cultural impact of the snackification of the diet cannot be overstated. We have moved from a society of 3 distinct meals to a society of grazing. This constant intake of food means the liver never gets a break. In a fasted state, the liver can engage in autophagy, a cellular spring cleaning process where it breaks down damaged cells and misfolded proteins. This is a crucial anti-cancer mechanism. However, autophagy is suppressed by insulin. By snacking continuously on ultra-processed foods, we keep insulin levels chronically elevated, effectively turning off the liver's self-repair mechanism. We are denying the organ the downtime it needs to heal the micro-injuries accumulated during the day. To lower the risk of liver cancer, a rejection of ultra-processed snacks is essential. This is not merely a matter of weight loss, it is a matter of toxicological defense. It requires a return to real food, food that rots, food that has a single ingredient, food that does not require a chemistry degree to understand. It means recognizing that a potato chip is not a vegetable. It is a starch-fat-salt matrix designed to exploit your biology. The convenience of these foods is paid for with the currency of cellular health. Every crinkle of a snack bag is the sound of an industrial supply chain that prioritizes shelf stability over human longevity. The liver, silent and resilient, bears the weight of these chemical intrusions until the burden becomes too great.
And the cellular order collapses into the chaos of malignancy.
8. High-salt foods. Pickles, salted fish, instant noodles. Salt, or sodium chloride, is a paradox of human biology. It is the mineral of life, essential for nerve transmission, muscle contraction, and fluid balance. Without it, we die.
Yet, in the quantities consumed in the modern industrial diet, it becomes a slow-acting poison, a corrosive agent that fundamentally alters the hemodynamics and cellular integrity of the liver. While the public health narrative on salt has largely focused on the heart and blood pressure, the investigation into liver cancer reveals that sodium plays a specific, mechanical, and devastating role in the progression of hepatic disease. The entry on high-salt foods is not just a warning against the salt shaker, it is an indictment of a preservation method that has outlived its necessity and evolved into a culinary addiction. We must look specifically at the trinity of high-sodium hazards: traditional salted preserves, the modern instant noodle, and the hidden sodium in restaurant fare. The relationship between salt and the liver is rooted in the physics of osmosis and pressure. The liver is a highly vascular organ, receiving 25% of the cardiac output. It acts as a blood reservoir. When sodium intake is excessive, the body retains water to dilute the blood, increasing total blood volume. This places immense hydrostatic pressure on the delicate sinusoidal channels of the liver. In a healthy liver, this is manageable. However, in a liver that is already compromised, perhaps by early-stage fatty liver or mild fibrosis, this increased pressure exacerbates portal hypertension. High pressure in the portal vein is the hallmark of advancing liver disease. It forces fluid to leak into the abdominal cavity, ascites, and causes varices, enlarged veins, to form. But at a cellular level, high sodium concentrations induce osmotic stress. This stress forces liver cells to swell, hypertrophy, and alters their cytoskeleton. Research indicates that mechanical stress activates the hepatic stellate cells, the architects of scar tissue. In the presence of high sodium, these cells produce more collagen, accelerating the transition from simple fibrosis to irreversible cirrhosis. However, the cancer risk associated with high-salt foods is most vividly illustrated by the consumption of Cantonese-style salted fish. This specific food item holds a grim distinction. It is classified by the IARC as a Group 1 carcinogen.
While primarily linked to nasopharyngeal carcinoma, the mechanisms of its toxicity are highly relevant to the liver. The traditional method of preparing this fish involves salting raw fish and allowing it to ferment under the sun. This process allows bacteria to convert the nitrates in the fish and the salt into nitrites. These nitrites then react with the abundant amines in the fish protein to form N-nitroso compounds, nitrosamines, specifically N-nitrosodimethylamine, NDMA. As previously established in the processed meat section, NDMA is a potent hepatotoxin. When this fish is consumed, often as a savory addition to rice or congee, the liver is bombarded with these preformed carcinogens. In populations where this is a dietary staple, the synergistic effect of viral hepatitis and salted fish consumption creates a cancer hotspot. It is a tragic example of how a survival technique, preserving protein in the absence of refrigeration, can become a generational health crisis. Then we arrive at the modern industrial incarnation of the salt bomb, the instant noodle. A single brick of instant noodles, with its accompanying flavor packet, can contain over 2,000 milligrams of sodium, nearly the entire recommended daily limit for an adult. But the sodium is only part of the equation. Instant noodles are typically flash-fried in palm oil to dry them out, creating a product that is high in oxidized saturated fats, entry number 5, and massive amounts of salt, entry number 8. Furthermore, to prevent the fat from going rancid, manufacturers add tertiary butyl hydroquinone, TBHQ. TBHQ is a synthetic antioxidant derived from petroleum. While FDA approved in small amounts, studies suggest that chronic exposure to TBHQ can enhance the car's enlargement and neurotoxic effects. The instant noodle diet, common among college students and low-income populations worldwide, is a perfect storm for liver dysfunction. High sodium causing osmotic stress, oxidized fats causing inflammation, and synthetic preservatives challenging detoxification pathways, all devoid of the protective fiber and antioxidants found in fresh food. The insidious nature of salt lies in its ability to mask the quality of food. High levels of sodium can make low-quality, degrading ingredients taste palatable. This is the secret of the processed food industry. But beyond taste, salt affects the structural integrity of the liver matrix.
Recent studies have shown that high salt intake leads to a decrease in the production of nitric oxide, NO, in the liver. Nitric oxide is a vasodilator. It keeps blood vessels open and flowing. When NO levels drop, the hepatic microcirculation constricts. This ischemia, lack of blood flow, causes localized hypoxia, lack of oxygen in liver tissues. Hypoxia is a well-known trigger for cancer. Cells that are starved of oxygen often mutate and switch to anaerobic metabolism, the Warburg effect, a characteristic of cancer cells. Thus, chronic high salt intake creates a microenvironment, hypoxic and fibrotic, that is favorable for tumor growth. We must also address the pickled category. Pickling is essentially a controlled decay using brine. While fermented foods like kimchi and sauerkraut have probiotic benefits, they are also exceptionally high in sodium. In Asian epidemiology, high intake of pickled vegetables has been consistently linked to higher rates of gastric and esophageal cancers, but the hepatic burden is often overlooked. The sheer volume of sodium required to inhibit bacterial growth means that a heavy consumer of pickles is chronically overloading their sodium-potassium pumps. This electrolyte imbalance forces the liver cells to expend excessive ATP, energy, to maintain homeostasis, depleting the energy reserves that could otherwise be used for DNA repair. The cultural dimension of salt is profound. In many cuisines, salt is not just a seasoning, it is the foundation of flavor. Soy sauce, fish sauce, miso, oyster sauce, these are the pillars of umami, yet they are liquid salt concentrates. A tablespoon of soy sauce can contain 1,000 milligrams of sodium. In the Western diet, the salt is hidden in bread, cheese, and sauces. The salt wars in public health have raged for decades, but the consensus is shifting toward the realization that our physiological tolerance for sodium was set during the Paleolithic era, where sodium was rare and precious. Our modern environment, where salt is ubiquitous and cheap, is an evolutionary mismatch. The liver, which regulates blood volume and filtration, is the primary victim of this mismatch. Moreover, high salt intake is a driver of autoimmune activity. It induces the differentiation of T helper 17, Th17 cells, which are highly inflammatory. In autoimmune liver diseases like primary biliary cholangitis, PBC, or autoimmune hepatitis, excess salt acts like gasoline on a fire,
accelerating the destruction of liver tissue. Even in those without autoimmune conditions, this pro-inflammatory shift weakens the immune system's ability to conduct surveillance, the process of identifying and eliminating early-stage cancer cells. The future of liver health depends on a palate reset. The human taste for salt is adaptable. If one reduces salt intake gradually, the taste buds adjust within weeks and natural flavors become more pronounced. However, the food industry resists this, knowing that salt is the cheapest way to ensure repeat purchase. To lower liver cancer risk, one must become a label detective, scrutinizing the sodium content of every package. It requires a shift away from preserved foods back to fresh foods. It involves using herbs, spices, citrus, and vinegar to flavor food instead of relying on the crude hammer of sodium chloride. Ultimately, the high salt diet is a form of physiological harassment. It keeps the liver under constant pressure, literally and metaphorically. It stiffens the tissue, constricts the blood flow, and accelerates the scarring process. By reducing sodium intake, we lower the hydrostatic pressure in the portal system, soften the liver tissue, and restore the delicate balance of fluids and electrolytes that is the prerequisite for cellular health. Avoiding the salt trap is a critical step in de-escalating the biological war being waged against our own organs.
Number 9. Excess red meat, fatty beef, pork. The consumption of red meat is one of the most polarizing topics in modern nutrition, yet when viewed through the lens of hepatic pathology, the debate shifts from ideology to biochemistry. For millennia, red meat was a rare, nutrient-dense prize that fueled the expansion of the human brain. Today, however, it has become a daily commodity, produced on an industrial scale and consumed in quantities that our metabolic machinery was never designed to handle. The investigation into the link between red meat and liver cancer requires us to separate the nutrient from the dosage. While beef and pork are rich sources of protein, B vitamins, and zinc, they also contain a trifecta of compounds that in excess act as architects of liver destruction: heme iron, saturated fats, and a unique sugar molecule known as Nu-5GC. Let us first examine the iron paradox. Red meat is distinguished from white meat by its high concentration of myoglobin, the protein that holds oxygen in muscle tissue. At the core of myoglobin lies heme iron. While iron is essential for life, it carries oxygen in our blood, it is also a volatile element. The human body has no active mechanism to excrete excess iron. We absorb it, but we cannot easily get rid of it. The liver is the body's primary vault for iron storage. When we consume excessive amounts of red meat, the liver accumulates high levels of heme iron. In this environment, iron acts as a pro-oxidant. It catalyzes the Fenton reaction, a chemical process where iron reacts with hydrogen peroxide, a natural byproduct of metabolism, to produce hydroxyl radicals. These are the most destructive free radicals known to biology. They rampage through the liver cells, shredding lipid membranes and smashing DNA structures. This state of chronic oxidative stress is a primary driver of fibrosis. In patients with hereditary hemochromatosis, a genetic inability to regulate iron absorption, liver cancer is almost inevitable without treatment. Consuming excess red meat mimics this condition on a lower, chronic scale, slowly saturating the liver with a metal that rusts the organ from within. The second component of the threat is the lipid profile. Modern grain-fed beef and pork are vastly different from the game animals our ancestors hunted. Feedlot animals raised on corn and soy and restricted from movement develop intramuscular fat, marbling. This fat is highly saturated, specifically rich in palmitic acid.
When ingested, palmitic acid triggers a specific type of cellular distress known as endoplasmic reticulum (ER) stress. The ER is the organelle responsible for folding proteins. When overwhelmed by saturated fats, the ER malfunctions, sending out distress signals that trigger inflammation and insulin resistance. This is the biochemical root of non-alcoholic fatty liver disease, NAFLD. The liver, unable to process the deluge of saturated fat, begins to store it. But unlike subcutaneous fat, liver fat is lipotoxic. It poisons the cells, leading to cell death, apoptosis, and the compensatory proliferation of scar tissue. As we have established throughout this investigation, the road to liver cancer is paved with scar tissue. Furthermore, we must explore the Neuro5-GC hypothesis, a fascinating and disturbing evolutionary mismatch. Most mammals produce a sugar molecule called N-glycolyl-neuraminic acid, Neuro5-GC, on their cell surfaces. Humans, due to a genetic mutation roughly 2 million years ago, do not. We produce a slightly different version called Neuro5-AC. However, when we eat red meat, muscle from mammals like cows and pigs, we ingest Neuro5-GC. Because our bodies do not make this molecule, our immune system recognizes it as a foreign invader, a xenoantigen. We develop antibodies against it. The problem arises because Neuro5-GC can be incorporated into our own tissues, including the liver, from the diet. When our antibodies attack the Neuro5-GC that has embedded itself in our own cells, it creates a state of chronic low-grade inflammation termed xenocellulitis. This persistent immune attack on our own tissues is believed to contribute to tumor formation. The liver, as the primary site of metabolism for these dietary sugars, is on the front lines of this immunological civil war. The investigation also leads us to the gut microbiome and the production of trimethylamine N-oxide, TMAO. Red meat is rich in L-carnitine and choline. When these nutrients reach the gut, specific bacteria convert them into trimethylamine, TMA. The TMA is absorbed and travels to the liver, where it is oxidized into TMAO. High levels of TMAO are not only linked to heart disease, but are also associated with liver cancer. TMAO disrupts bile acid metabolism and promotes hepatic inflammation. It creates a toxic microenvironment that supports the survival and growth of malignant cells. This highlights that the danger of red meat is not just in the meat itself,
but in how our internal bacteria interact with it to produce secondary toxins. We must also consider the method of preparation, which often compounds the risk. Red meat is frequently consumed as processed meat, entry number 3, or charred meat, entry number 4, meaning the consumer is hit with a triple threat of heme iron, nitrates, and heterocyclic amines. But even unadulterated fresh red meat poses a risk when the volume is high. The World Cancer Research Fund recommends limiting red meat consumption to no more than 3 portions per week, about 12 to 18 ounces total. Yet in the Western diet, it is common for individuals to consume this amount in a single day. The 16-ounce steak, the double patty burger, the pork chop dinner, these portion sizes are normalized cultural artifacts that defy physiological limits. The shift in agricultural practices has exacerbated the issue. Cattle evolved to eat grass, which results in meat with a balanced ratio of omega-3 to omega-6 fatty acids. Grain feeding shifts this balance heavily towards omega-6s, which are pro-inflammatory. Thus, a modern steak delivers a high dose of inflammatory fats along with the heme iron. This inflammatory load keeps the liver in a state of constant reactivity. Over decades, this chronic inflammation exhausts the liver's regenerative capacity. The cells stop dividing neatly and start dividing chaotically, leading to the genomic instability that characterizes hepatocellular carcinoma, HCC. There is also a significant correlation with obesity. High red meat consumption is consistently linked to higher body mass index, BMI, and larger waist circumference. Visceral obesity is the single strongest risk factor for NAFLD. The fat tissue releases adipokines, hormones, that promote insulin resistance in the liver. It creates a vicious cycle. The meat contributes to the obesity makes the liver more susceptible to the meat's toxicity, and the liver loses its ability to regulate metabolism, leading to more obesity. The future of this issue is currently being written in laboratories. The rise of plant-based meat alternatives and cultured, lab-grown meat attempts to solve the ethical and environmental problems of animal agriculture, but the health implications for the liver remain to be seen. If lab-grown meat replicates the heme iron and saturated fat profile of conventional beef exactly, it may carry similar risks. Conversely,
plant-based analogs often use coconut oil, high in saturated fat, and isolated proteins which present their own processing challenges. The safest path for the liver remains a shift toward a plant-forward diet, where red meat is treated as a garnish or a rare ceremonial food rather than the centerpiece of daily nutrition. Ultimately, the liver is a forgiving organ, but it is not invincible. It can handle the occasional influx of heme iron and saturated fat. It cannot, however, withstand a daily barrage. The correlation between high red meat intake and cancer is a signal that we have drifted too far from our evolutionary baseline.
By reducing red meat consumption, we lower the iron load, reduce oxidative stress, dampen inflammation, and deny the cancer process the fuel it needs to ignite. The carnivore instinct must be tempered by the reality of the oncological consequence.
10. Raw or unsafe seafood, unclean shellfish. The final entry in our investigation brings us to the intersection of gastronomy and microbiology. The consumption of raw seafood, oysters, clams, sashimi, ceviche, is often celebrated as the ultimate expression of culinary purity. It is the taste of the ocean, unmasked by heat or spice. However, this pursuit of freshness carries a distinct and lethal set of risks for the liver. Unlike the previous entries, which cause cancer through metabolic toxicity or chemical carcinogenesis over decades, raw seafood acts as a vector for biological agents, bacteria, viruses, and parasites that can attack the liver with terrifying speed or set up a chronic residence that leads to malignancy years later. In a world of warming oceans and compromised water quality, the raw oyster is not just a delicacy, it is a bioaccumulator of environmental hazards. The most immediate and catastrophic threat comes from Vibrio vulnificus. This is a bacterium that thrives in warm, brackish coastal waters. For a healthy person, ingesting Vibrio might cause a nasty case of gastroenteritis, but for a person with pre-existing liver disease, such as cirrhosis, hemochromatosis, or chronic hepatitis, Vibrio is a death sentence. The liver is part of the body's immune defense, containing cells that filter bacteria from the blood. In a compromised liver, this filtration system fails. Vibrio moves from the gut into the bloodstream and multiplies with explosive speed, causing septicemia, blood poisoning. The mortality rate for liver patients infected with Vibrio vulnificus can exceed 50%, with death occurring within 48 hours of symptom onset. While this is not cancer per se, it is a liver-specific fatality linked directly to diet. However, the chronic inflammation from repeated, sublethal infections can accelerate the progression of existing liver disease toward cancer. The more direct carcinogenic link lies in the realm of parasites, specifically liver flukes. The species Clonorchus sinensis and Opisthochus vivarini are parasitic flatworms found in freshwater fish across East and Southeast Asia. When humans consume raw, undercooked, or pickled freshwater fish, a common practice in dishes like koi pla in Thailand or raw carp in China, they ingest the infective larvae. These larvae migrate from the intestine up the bile duct and into the liver. There, they mature into adult worms and can live for decades.
Feeding on bile and causing chronic mechanical irritation to the bile duct walls. This is not a rare occurrence. Tens of millions of people are infected worldwide. The presence of these flukes triggers a massive, persistent inflammatory response. The cells lining the bile ducts, cholangiocytes, proliferate in an attempt to repair the damage caused by the worm's suckers and metabolic waste. Over time, this chronic proliferation leads to DNA mutations. The result is cholangiocarcinoma, bile duct cancer. This is a rare cancer in the West, but a major epidemic in regions where raw freshwater fish is a staple. It is an aggressive, often fatal malignancy that is almost entirely preventable by cooking fish. The tragedy is that the cultural tradition of eating raw fish is so deeply ingrained that public health campaigns struggle to change behavior, even as the cancer wards fill up. We must also investigate the viral threats, hepatitis A and hepatitis E. These viruses are transmitted via the fecal-oral route, often through the consumption of shellfish harvested from waters contaminated with sewage. Oysters and clams are filter feeders. They pump gallons of water through their systems to extract plankton. In doing so, they concentrate pathogens in their tissues to levels far higher than the surrounding water. If you eat a raw oyster, you are essentially eating the concentrated biological content of the harbor it came from. Hepatitis A causes acute liver inflammation. While it usually resolves, in older adults or those with underlying liver conditions, it can cause fulminant hepatic failure. Hepatitis E is particularly dangerous for pregnant women and immunosuppressed individuals. While vaccines exist for hepatitis A, they are not universally used, and the global food trade means that contaminated shellfish can appear on plates thousands of miles from the source. The environmental context of this risk is darkening. Climate change is warming the oceans, expanding the range and season of Vibrio bacteria. Waters that were once too cold to support high bacterial loads are now becoming incubators. Furthermore, harmful algal blooms, red tides, are becoming more frequent. These algae produce hepatotoxins like microcystins and nodularins. Shellfish accumulate these toxins. When humans consume them, these toxins target the liver, causing acute damage and promoting tumor growth through the inhibition of protein phosphatases, enzymes that regulate cell division.
We are witnessing a synergy between environmental degradation and dietary risk. As we pollute and warm the oceans, the food we pull from them becomes increasingly weaponized against our livers. The definition of unsafe also extends to the heavy metal content of large predatory fish. Mercury, while primarily a neurotoxin, also accumulates in the liver. High levels of mercury induce oxidative stress and deplete glutathione, the liver's master antioxidant. This leaves the liver more vulnerable to other carcinogens. Tuna, swordfish, and shark, often eaten raw as sushi, are top-tier predators that bioaccumulate mercury. Regular consumption of these raw fish contributes to a toxic heavy metal load that stresses hepatic detoxification pathways.
The cultural allure of raw seafood is powerful. It suggests sophistication, connection to nature, and health. The oyster bar is a symbol of high society. However, the biological reality is that eating raw shellfish is an act of gambling. Lemon juice and hot sauce do not kill bacteria or viruses, only heat does. The R rule, only eat oysters in months with an R, is an old wives' tale that no longer holds true in an era of global warming and global supply chains. For the vulnerable, those with fatty liver, viral hepatitis, or heavy alcohol use, the advice must be absolute. Avoid raw seafood. The compromised liver cannot handle the bacterial challenge of Vibrio, nor can it afford the additional insult of viral hepatitis or parasitic invasion. For the healthy population, the risk is lower but never zero. The investigation reveals that the ocean is not a sterile country. It is a complex, living ecosystem teeming with organisms that view the human liver not as a master organ but as a host. To lower liver cancer risk, we must reevaluate our relationship with the raw. Cooking seafood is the single most effective intervention to neutralize these biological threats. It kills the Vibrio, it kills the virus, and it kills the parasite. It transforms a potential biohazard into a safe source of nutrition. In the grand calculation of risk, the texture of a raw oyster is a poor trade for the integrity of the liver. We must recognize that in the modern world, fresh does not mean clean, and the most dangerous thing on the plate may be the one that is still biologically active. This concludes the investigation into the 10 dietary assassins of the liver.
From the solvent properties of alcohol to the microscopic invasion of liver flukes, the threats are diverse, yet they share a common mechanism. They overwhelm the liver's capacity to protect itself. The liver is the guardian of the body, but it requires a guardian of its own. That guardian is the conscious, informed choice of what we allow to pass our lips. By avoiding these 10 categories, we do not just avoid disease, we actively cultivate the resilience required for a long and vital life.